then hopefully you can see it, it's not too bright and so on. So I will start to focus on the taxa that's commonly occurring in the Baltic Sea proper, and the nomenclature and also the taxonomy that I've used here is based on a report written by Guy Helfors, and it was published 2004, and it's a compilation of all the information about what species have been found in the Baltic Sea area, from Kattegat into the lagoons and so on. So, and this has been a lot of changes. As you know, when working with taxonomy, it's not an easy task. There is a lot of things going on, and nowadays you have more molecular methods too. And then you can realize that you have to split this species or this group into 5 or 6 or many more, and there is a lot of things going on. So it might not always be the latest taxonomy used here, but I wanted to be a bit more consistent, so I use this from Guy Helfors. And that is also what we are using when we are working with monitoring samples of phytoplankton. We're basing it on this, and also on this other paper called Cell Sizes of Baltic Sea Phytoplankton. There is a group of phytoplankton experts around the Baltic that are gathering once a year, and we're going through this list of species, and also looking at different size classes of the different species, so that we're using standard size classes, so that we're differences in biomass is not due to that we are using different formulas and other things, so that's standardized. And these are the main working documents for us within the monitoring program. And if we look at the classification of phytoplankton, we have from the highest division, class, order, family, genus, genera, and the species. And if you want to look this, there is also a classification if you're using for the zoological theme. Then there is another ending of the division and the orders and so on. But this is the floral way of naming species. And the division is ended by phyta. The class is ended by physe, and the order ales, family not ase, and then we have the genus and the species name. That's basically, you have seen this with all the other groups, I suppose, so it's nothing new, but it's easier to find where you are in the taxonomy, because when you're looking at phytoplankton and other things, it's not always you can use the species name. Sometimes you have to use the family name, or sometimes even the order or class, because it's difficult to say what species it is. So sometimes you use the class or the order in monitoring too. So then it's good to know which one it is. And a bit short, we're looking at the smallest things here now, and we have divided the phytoplankton or plankton in different size classes. We have the picoplankton, they are smaller than 2 microns. We have the nanoplankton, 2 to 20 microns. Microplankton, 20 to 200. Here starts the zooplankton coming in, the ciliates and other things, and mesoplankton, that's the larger phytoplankton, and of course the zooplankton that you were looking at yesterday. And also we have different trophical grades in phytoplankton. We have the autotrophs, they are using photosynthesis for their carbon, and so we have autotrophs. We have those that cannot use photosynthesis. They have to eat other particles. They are the heterotrophs. And then we have the mixotrophs that can use either they can shift between being autotrophs or heterotrophs, and they're both. So we have it. And in this compendia, I have tried to write down if they are autotroph, heterotrophs, or mixotrophs, where possible. In some groups, they are all autotrophs, and there I haven't written anything about it. So AU is autotrophic, HT heterotrophs, and MX is mixotrophs. So you know in the compendium we're looking at it. And then a quick overview of the larger divisions we have of the Baltic phytoplankton. We have the Cyanobacteria. You have probably seen them as threads in your samples yesterday. And we have the Cryptophytes, they're usually small things. Dinoflagellates, Haptophytes, Chrysophytes. Here you have the Diatoms, Euglenoids, and Chlorophytes, including the green algae. Then we have also some autotrophic or mixotrophic ciliates that are also important for the Baltic. It's mainly one species, and I will talk about that. And there are also some zooflagellates. They are heterotrophs that you can see a lot also in your samples, and I will talk briefly about those two and just show them what they are. As you can see, this is just a summary for you. There are a lot of classes and orders under all these divisions, and I will go through all these groups and talk a bit what's specific for the different groups. And so this is just, I made this compendium so it's something that you can go back to if you want to look more at phytoplankton, also in the afternoon. Then you can go and look, what is this and what group is it and what species might it be, and so on. So hopefully it's useful to use the compendium. And also in the end of the compendium there are a couple of references for good taxonomical books and so on. If you are more interested in phytoplankton, you can find some more information there. And the continuation of what we're going to look at today. And we will start with the Cyanobacteria, and the class is Gnosticophyse, and before they were called also, you have probably heard blue-green algae, but nowadays they always say Cyanobacteria. And Cyanobacteria, they are very much like, they are something in between bacteria and eukaryotes, autotrophs. They are not eukaryotes, they have no cell carrion, and all the material in the cells are spreaded in the cells, so they are not specific areas for things. So, but there are a lot of autotrophic cyanobacterial species in the Baltic, and they contain a lot of pigments like the algae, and they can have from green to blue-green to red color. When I went by train today from Stockholm, I saw when we were crossing the bridges at Skanstull, the water is green. What is it? It's a cyanobacteria bloom of something in Lake Mälaren there, so it was quite nice. Actually, I wanted to stop and take a sample, but that's me. But they're quite fun there to look at, but they are more freshwater species, and I'm not that acquainted to all of them, so that's something interesting for me to learn too. Okay, but some of them are toxin producing, and that's why you can see some signs in the newspapers during summer times that you should not go out, take a swim when there are heavy blooms of cyanobacteria blooms. And also some of them, or a lot of them, contain small gas vacuoles or gas vacuoles, which make that they can regulate their where they are in the water column. And usually when they are getting old, this regulation is not working, and they are usually accumulating on the surface. So usually what you see, the surface accumulation is usually old blooms of cyanobacteria. And they are mainly occurring during summertime in the Baltic. Of course, there are small cyanobacteria that can be occurring all year around, and also some of the other species too. Then we will look a bit at the groups. There are 3 large orders of cyanobacteria: Chrysophytes, Oscillatoriales, and Nostocales. And I will go through these, and you will see some pictures of these, and you will see, just to make you see the difference about how different they can look to. And we will start with the Chrysophytes. They are unicellular. They're very small. Usually the sizes are less than 1 micron, or between 1 and 2 microns. So they were very small, and they have, due to that, they have very effective nutrient uptake. They are usually occurring then they are abundant during summertime, and they can take up nutrients that other phytoplankton are leaking. And some of them are forming colonies because they have mucilage around them. So either they can be single cells, like this Synococcus type, this is from a culture, or they can be colony forming like this Aphonotheca. So it had a rigid structure. And these are usually not the single cells are not visible in the water, but the colonies can be quite big, so you can see them in the water as grains or particles, and that is probably what I've seen today. It's some gelatinous colonies that I could see from the train. This is just to summarize this order, and we have some of the most common genera. We have this Aphanocapsa. They are, when you're looking at them, it looks more like a cloud with small dots on it. And what's striking with this genera has round cells. So the cells are round, and the cells are very small. This one has cells that are about half a micrometer or something in size, but they can be very abundant in them. And also, if you're counting this, it's very tedious if you have to count all the cells in the colony. So usually you're approximating how much you have in one field, and then you're counting, for example, units of 100 cells, and you know that this part of the colony is 100 cells, and then how many of those you have in a colony, and then you try to approximate. But for these, I can say it's quite arbitrary. It's not that easy to get a good biomass estimate, so it's not the easiest. Then we have the Aphonotheca. They are more elongated, the cells, and I like this species because this is one of the cyanobacteria that you easily can say it's this species because it looks like nothing else. The cells are in rows. Usually the colonies are quite flat, and they're very nice to see. When I was starting as a PhD, this was actually not described, and I was on one of these meetings they have within this HELCOM phytoplankton group, and in all samples they were looking at during a course of cyanobacteria, they had this, and I asked, what is it? Do you know what to call it? Because I have a lot of it in my samples too, and I thought it was described, but it wasn't. But then a couple of years after, it was described by one from Lund University. So it was nice to have a name of something that you can see actually this is one of these. So I like that one. So it's easy because it can be sometimes difficult to see if they are round or if they are elongated. So it's not always easy. So there are a lot of different. And then we have another very common generates this Cyanodictyon. They have the colonies can be very large, and they have also elongated cells. But the cells are usually also in rows or in pairs. And so there is some kind of structure, some net structures in the colonies. So usually you look for roundish or elongated sails and some net structure in it. And then it's usually this cyanodictyon. Then you have cyanonephron. It's looked like kidney-shaped, the cells a bit more. And the colonies are usually not that big, so. And we have this lamellariella. It looks like a football with all the cells on the outer part of the cells, so they can be quite nice. There are two species here, one with round cells and one with elongated cells. And we have this Meresmopedia. We have a couple of species occurring in the Baltic, but there are more species in freshwater. And they are usually sitting 4 and 4 cells together, and it can be colonies. They're usually flat colonies, like a paper of cells. And they're always arranged 4 and 4 together. So they're quite nice to see, and there you use the sizes and the forms of the cells to distinguish between the different species. And then we have the microcystis species. Most of these are usually more freshwater species. So what I saw from the train was probably a mixture of these species, I suppose. And many of them are toxic too. So we have this Microcystis aeruginosa. It has small gas bubbles in it, so you can see small dots in the cells. You have this Microcystis viridis. It look like, if you're looking at in the microscope, they look like grapes on a whole bunch of grapes. And you have this Microcystis wesenbergi with these gelatinous sheets that makes it quite easy to distinguish them from something else. So these are very common in a lot of lakes and so on. You can make real problems if you're using the lake for water supply. So they have had some real problem in Lake Ringsjön and Maddersjön in Skåne area. And this is two common groups in the Baltic. You have Snovella. It's loose colonies with gelatinous threads connecting the cells. And then you have this Voronezhinia. It's more compact, the colonies. There should be some kind of gelatinous stalks inside too, but they're usually very difficult to see. You don't see them in it. And the cells are usually arranged on the outer part of the colony like this. And you will probably see these in the samples later in the afternoon. And then we have Oscillatoriales. It's filamentous species of Cyanobacteria, but without heterocytes, and they have no nitrogen fixing possibilities. They have no nitrogen fixing cells, at least what we know. Of course, maybe Isabel and other might have some more to add to that in the later if they come up with some more interesting about your. So we'll see if you can find if they can fix somewhere else in the cells. And they are always unbranched filaments, and this genus Pseudonomena is very common in the Baltic, and it occurs during summertime. And this Planktotrix is one that can be also occurring in lakes, but also in coastal areas, and it can also be very toxic, and it can color the water red if it's in large quantities. And just to see what they look like, we have a couple of common genera here too. We have Planktolymbia. Here the cells, it's a long thread in a gelatinous sheet. Here you see that the cell has divided, or the filament has divided, but there is still this sheet around it, so it's connecting the cells together. So if you see these empty parts, you can see that it's belonging to this Planktolymbia part. And they can be either straight or like this Comporta, it's a curled species. And this is, for example, very common in the coastal areas with high nutrient concentration. You can have a lot of these. This pseudonabena is common here in Askö and Himmelfjärden area here around during summertime. This Planktotryx that I talked about, it has small gas bubbles in it, so you can see you can see some structure in the filaments, and this is one species that can be toxic. I suppose the toxicity has more been found in lake samples, but it can be toxic in the Baltic too. And then we have the Nostocalis. That's the most interesting part, maybe, of the cyanobacteria, if you're looking at Nodellaria and Aphanisomnon, because here is where you find them. They are also filamentous, and they have these heterocytes and actinides. Heterocytes are the nitrogen-fixing part of the colony, so they can fix nitrogen during summertime, where there are low amount of nutrients or nitrogen available in the water. And they have actinides, that's resting spores. So at the end of the season you can see increasing amounts of actinides. I saw some of them from last week in the sample, so maybe we can see them also in the live samples you took yesterday. Otherwise I have some that you can look at. And some of them, like Aphanisomnon, they are small filaments, but they are creating bundles, tight bundles and colonies, so you can see them in the water. If they are just one filaments, you wouldn't see them, but since they are a lot of filaments together, you will see them as green sticks in the water. So Aphanisomnon is the most common cyanobacterial species in the Baltic. More. And I will go through the most common groups here. Maybe you don't know this genera, but Dolichospermon was before called Anabena. All the Anabenas, or this group, or this genera has been divided into two genera now. All the benthic species are still Anabena, but the pelagic species are now called Dolichospermon. But in our bi-volume list, the list that we're using for monitoring, we still use Anabena, but we're going to change it for next year, but it will be changed. But there are a lot of things going on in this too, so now I have changed it. So in this Hellfors, it's called Anabena. And there were 8 different species in here, and you can, for species identification, you see the cells sizes, the form of the cells, and this is the heterocyte, and around the darker ones are the actinides. And how the cells and the actinides and the heterocytes are arranged together, it can also be used for identification of the species. So this Dolichospermon lemmermanni, it's special that it has the actinides around the heterocytes. Here all the actinides have gathered in the middle of the colony. But they look like pearls on a string, then, this Dolichospermon group. And then we have this Aphanisomnon. In the water they look like green sticks, and they're just single filaments. Here you have one single thread. You have the heterocyte, the vegetative cells, and then you have the actinides, or the resting spores. And this is the most common species during summertime. And also this species can be occurring all year around in small amounts, so you can find it, especially in the open Baltic, maybe not always in coastal areas, but you can find it all year around. So it's thriving in maybe if it's around 15 degrees, then it can start growing a lot compared to other species. And then we have these two are not that common, but they can be occurring in coastal areas. But I just wanted to show that the heterocytes can be found in different areas, or the form can be. This Anabenopsis, it has the heterocytes on the end of the colonies, and it's also curved, the colony. But in this Kronbergia instead, they have the heterocytes on the side. They look very much like the Anabenopsis, but they are straight, the colonies instead. So how they look like can be a way to identify the group. And then we have Gliotrichia. It's mostly occurring in lake areas, and this is common in Lake Mälaren, and this is probably what I saw also from the train. And it's like a ball of threads, like I don't know, in Sweden we use, when I was small, you were doing a ball of garn, as the one you have on your hat or whatever it is. They look very much like that, so it's tight in the middle, and usually the heterocytes are gathered in the middle, and then you have the vegetated cells going out in straight lines like this. And the colonies can be as large, you can see them as green balls in your hand if you take them. They're a couple of millimeters in size sometimes. And then we have the Nodularia spumigena, the Nodularia. There are a couple of species in the Baltic, but the most common is Nodularia spumigena. And it has, as the others, they have the heterocytes, and it's almost evenly spaced in the filament. You can see the brighter part are the heterocytes, and in between you have the vegetated cells. And the colonies can be either straight, they can be curled, and they can be like a rubber band that you're twisting and then losing it, and then it's very difficult to count this. If you want to count, you usually count the length of the filamentous species. And you can imagine if you have a lot of these, you're not that happy, because it takes some time just to, how large is it, approximating. And also these, if you have a very curled one, you can have very tightly curled filaments too. And it's a bit tedious to look at them, so I prefer the straight ones. And then we go to the next group, is the Cryptophytes. They are very small things, and you will not be able to see them one by one in the water. They're usually in the size from 4 microns to maybe 30 microns in size. And they are called also recoiling algae. They are always unicellular, and they have 2 flagella, and usually the cell is what's called dorsoventrally constructed. It has this part, the back, is convex, and the ventral part is more straight and flat. And in this part it has a small gullet, or svalg in Swedish, where the flagellas are attached somewhere here. And they have 1 to 2 chloroplasts and different accessory pigments. And usually they are having a lot of pigments that make them, that they can stay quite deep in the water. So you can find these at 20-30 meters depth, where there's not much light there. So they can utilize the light that is just penetrating as far as that. And that's why they have these, because they have these accessory pigments, and they can also migrate vertically to get a better nutrient area to thrive in. And they are an important part of this nanoplankton, this plankton between 2 and 20 microns, and they're important during the summertime and in autumn, but they are occurring all year round. And the most common genera are the Hemicelmus, the smallest one, the Plagiocelmus, the middle-sized one, and the Teleaulax. And in Baltic we have just one species, it's Hemicelmus virescens, and Plagiocelmus we have also one species, we think, at least for now. So, and we have 2 species in Teleaulax. And then we go over through to the Dinoflagellates. There are a lot of different Dinoflagellates, and they are usually unicellular and a few can form colonies, and I will show that too. And they have a special arrangement of the cells. And there are two important groups in Dinoflagellates. We have what's called the naked or the Euglenoid cells, and the armored and heterotrophs cells. The naked, they have no plates like the pyrenoid cells. They have what are called taxa plates, and these plates can be used for identification of these species. So how they are arranged, the size and the form of these, and how many plates you have are used for identification of these armored Dinoflagellates. And what's common with all the Dinoflagellates is that they have what's called epitheca or episome, the upper part, the head part, and then epicone or hipposome, the lower part. And in the middle they have a cingulum or a girdle, and they also have what's called a sulcus, a list, something going down here. And in this cingulum they have a flagella that is moving the cells around and round, and then for moving back and forth you have a flagella going down in this sulcus. So it's found also in this the taxa species too. And here you have the flagella that's going around the cells. And for identification, it's important to look at the shape of the cells, the position of the girdle. In this we have the girdle here. Here we have the girdle here. This one has a very big episome and a small hipposome. This one has a small upper part and a big lower pone. The hipposome is very small here. And in this one you have the girdle here. So the episome here is very small and a very big lower part, the hipposome. In this one it's more evenly distributed between the epi and hipposome. And it can have spines that can be used for identification, and it can also have some horns and these kind of spines. And also, of course, the taxa plate structure is important for their identification. And Dinoflagellates, they can be autotrophic, mixotrophic, and heterotrophic. Can be all. There are a lot of different species, and in Baltic there are all about described 200 different species. So it's quite a lot of different species. And the mixotroph, they can ingest bacteria, and when you're looking in the samples, they are moving around a lot. And also the autotrophs, they're also moving around, so sometimes it can be difficult to see, is this really a plant or is it some animal, because when they are moving around you think more it's like some zoological things, but it's not. And some of them, they can prey on very large diatoms. Sometimes you can see them eating large diatoms, or can find large diatoms in their cells too. So and also even polychaete and nematodes, polychaete larvae, so they can be hungry, the heterotrophs, but I haven't seen them eating this, but it would be nice to see them. Yeah, and also they have a different kind of accessory pigments, usually kind of carotenoids and pyridine. This makes them usually reddish brown, and that's why large blooms of Dinoflagellates are usually called red tides. And we don't really have so much red tides here, but in other areas it's very common to have red tides. And a lot of species are also toxic, and this group, Dinophytes, they are occurring on the Swedish west coast, and they are producing both this paralytic and diuretic shellfish poisoning. So when they are aggregated in the filtering mussels, and if you're picking those mussels, you can get really sick if you're eating them. So on the west coast, they are taking samples of this Dinoflagellates regularly during summertime, during the time when people are picking mussels, to be able to see if they are okay or not. And most of the Dinoflagellates are marine species, and as I said, around 200 have been reported from the Baltic. And they are also occurring year-round, but they're most commonly occurring during spring and summer, but it's different species and groups. And the heterotrophs are usually not that common. They're common during the end of spring bloom, but they are not usually more than a part of the biomass, at least here in the Baltic. And the Dinoflagellates, since they have flagellas, they can vertically migrate, and they, at daytime they are closer to the surface, and nighttime they can go down to the picnic line and look for more nutrients below that. So, and they have both sexual and reproduction in some of them, and a lot of them produce cysts too, and the cysts can be used for identification. Yes?
Do you have those fluorescent Dinoflagellates here as well?
Yes, there are some that can be bioeliminating. I will show you one that have been occurring in the Polish water, and also outside Gotland, and this the archipelago sea between Sweden and Finland too. And there are a lot of different orders. This is just for information for you, but we will look more at the most important orders for the Baltic, and it's these 5. And I just want to show you how different they can look like. And firstly, we have the Protocentrales and this genera Protocentrum. 2 common groups and species are Protocentrum minimum and Protocentrum micans. This is usually occurring more on the west coast, and you can see it sometimes up to Gotland Sea, usually not here. But this species is actually, it seems to be a more, they have called it an invading species, that it wasn't found in the Baltic before, and during late 80s or something it started to come into the Baltic, and some years around last 1997, 98, we had a lot of them here, and around 2000 when I was here with the course, there were a lot of them in the areas here around. But they're occurring some autumns and some not. They're usually, after the summertime they're usually occurring, and they can be very abundant when they occur. And they have this Protocentrum minimum, it has been found to be toxic in other areas. I don't know if anyone have found that it's toxic in the Baltic, but it is a species to be aware of if it's very abundant. And this species has 2 larger plates. On one side they are a bit flat on the side. If you see them from the side, it looks more like a straight, they're very flat, and they have some small plates around this area. Questions? And then we have this Dinoflagellates. It's this species that it has both mixotrophic and heterotrophic species in the Baltic. The most common heterotrophic species is this. It has been called Dinoflagellates rotundata, but it's also called Phalachroma rotundata, another genera. So I'm not really sure what it should be called now, if it's Phalachroma nowadays, I think. But other common groups are especially these Dinoflagellates acuminata and Dinoflagellates norvegica. These are the most common in the Baltic. This is actually occurring on the west coast, Dinoflagellates acuta. These they are all producing toxins, and they are monitoring them on the west coast. And this acuminata is quite roundish. It has, as I talked before, the upper part, the episome, is very small. You have the girdle already here. So the hipposome, the hypotheca, the lower part, is very big in this species. And this Dinophysis acuminata is quite roundish. This Dinophysis norvegica, it's usually thickest on the middle somewhere, compared to it's very much look like, and it has a bit invagination here usually. You don't always see it. And it looks a lot like this Dinophysis acuta, but this is more having the thickest part lower than the middle. It's looked like someone having a big belly and cannot really lift it usually. But it's more common on the west coast. And this species can also have, this norvegica, you can see a lot of structures on the list here. And usually you find them more at the end of the summer. And it might be something that they're getting the cells bigger to make them not as easy to eat from zooplankton and other things. So it might be something, or to stay up in the water column, or it's some kind of structure to adjust, because you can see more of this when there are a lot of zooplankton around, so it might be something like that. And then we have the naked species. These are two common genera, the Amphidinium and the Catodinium. This Amphidinium, they have a very small upper part, episome, this part, and a big lower part. Here you can see the nucleus of the Dinoflagellates. Compared to this Catodinium, you have a big, this is the episome, the upper part, and the lower part, the hyposome, is very small. So to see the difference between these genera, you should have a live sample, because you can see in what direction they are swimming. But in monitoring samples, when you have LUGO, you cannot see them swim at all. So then these three species are quite easy to distinguish between. Otherwise, you have to use either the genera, or even use Euglenoids if you cannot decide if it's either or when you're looking at them. And we have some more naked species. There have been a lot of Gymnodinium species. Now a lot of them have been redirected to other genera, because nowadays you have the molecular methods to decide what species it is. Because in the problem here, in these, you just have the size and the form of the cells, but you have no plates and other structures to look at, and they can look very much the same. So then the molecular methods are very nice to look at. And the Gymnodinium are more roundish, and this Gyrodinium, it's like two cones together. And this is a common heterotrophic species in the Baltic, and they can be big compared to everything else, can be around 100 microns in length. And in this one, you can see that this cingulum, or the girdle, can be distorted. It's not going back at the same place where it started. Here it's starting here, but then it's going back like that. So that's also a way of distinguishing between different species, how this distortion is, how big it is, and the length of that. It can be used for identification. And maybe Annika Sundström, one of our PhD students, she has been working on this complex of spring Dinoflagellates. And they look like, they are also called the meatball complex sometimes, because in monitoring samples they look like brown dots or brown meatballs, and they can be very difficult to distinguish between in the samples. but they are actually 3 different species, or I don't know actually if this is the names valid now, because this had been changing for the last 5 years back and forth for I don't know how many times. So now we call them, in the monitoring, we call this Scriptiella complex when we are counting it. And it has been 3 different species and also different orders. So this is a very difficult group, so it's not always easy to distinguish them. And they are common at the during springtime, and in some areas they are occurring even under the ice. They can start blooming under the ice and be very abundant just after the ice has gone through. It's common in the Finnish areas. There have been quite heavy blooms of this species. And it seems that we might not have the same species here than we have on the Finnish side, but hopefully Annika will tell us more when she's finishing her PhD soon, I hope. So it would be nice to see if it's the same species or not. And some of these other tickles that have these tickle plates, this is this Heterocapsa. We have a small species called Heterocapsa rotundata. It's occurring all the year around, and it's very small. It's 10-15 micron in length. It looks a bit like a mushroom with a smaller part in the hyposome and the bigger epitheca. And this species, Heterocapsa triqueta, it's common late summer and also in upwelling areas. So it's quite abundant here in Askö areas, because we usually have upwelling here in this area now and then. And there is a 3rd species. It was not mentioned in Helfos because it wasn't described then, but it is this Heterocapsa arctica subspecies frigida. It's a spring species too that are occurring during springtime, and we might see them in some of the samples that I took with me. And then we have a genera that's called Glenodinium, and it has been a wastebasket for whatever you don't know actually what it is. There have been species with very thin theca, or there have been unknown what to use. I don't really like to use this genera because they're usually something else. I try to use something else than Glenodinium because most of them should be something else. So. And there are 2 more. I didn't have any pictures of this. This Cryptopyridinium foliosum, I know there's a PhD student working with that from the Archipelago Sea, from the Finnish areas. But it's difficult to see in monitoring samples because you usually see it from one side. But if you see it in live samples, you see it very flat, actually. So when it's swimming, it's easy to identify it, but not in the monitoring samples. But then we have this heterotrophic group. It seems to be a complex of different species. There were this Oblea rotunda species, and they are very roundish, and you can see the list of the sulcus or the cingulum going around here. So it's very special. And it could look like a plum with this small, where they are growing on to the tree when you're seeing them in Lugol sample or preserved samples. And then you have different species that can have spines that like this. These are all heterotrophic, this Protopyridinium, and they are common. Can be occurring mostly around all year round, especially these 2, but mostly occurring at late spring and summertime. And I saw a lot of these in the samples from last week, so we'll probably see them from the samples you took yesterday. Maybe swimming too. I tried to identify it, but it couldn't stay put. It was always swimming around because some of these, this one looked like it has trousers on, if you're imagining like something it's going up here. So it's, and this one, especially with this one, is it's this list of the sulcus is making like a third spine here. But if it's swimming, you don't really see it. And if you're monitoring samples, they're usually not lying in that direction, so you can see that. So it's not easy always to identify it. But that's one of those. And here we come to one of these bioluminescent species. This Alexandrium group, there are a lot of toxic species in this group. And this Alexandrium ostenfeldi has been found in Gdansk Bay a couple of years ago with bioluminescence, and also outside the island of Gotland, and also in the archipelago sea. So, and I haven't seen it, we have seen a few cells here too, but I've never seen bioluminescence, but it would be nice to see it. So if you're here seeing it, please take a samples. So it would be nice to see. And then we have this Amylax creacantha. It's just one species. It has special big horn, and this is not what I know, not the toxic species. It's occurring during springtime. And when we have the Ceratium species, they are mainly marine species. Especially, this is some examples of the marine species. The Ceratium tripos. They can cause red tides because they can color the water red. I don't think they are toxic, but if they are very high abundant, they maybe can be difficult for fishes and other things because they have these long horns. And we have one, this is an example of a freshwater species, Ceratium pyridinoella, that can be occurring in coastal areas. And then we have more Dinoflagellates. They are, as I said, we have 200 species there, can be very different. This is a group that has spines, the Cladopixus. There's 2 species, one with roundish cells, and this should have a bit more pointed episode. So, but it's common for this genera that has these spines. I think it's the only one in the Baltic that have these long spines, so it's quite easy to understand, to identify it. Then we have different kind of Garneolax species, and they can have different lengths of the spines to use for identification. And the problem with these are when they are fixed with Lugo that we're using, an iron solution, they're usually very dark. So it's very difficult to identify them. So you really need to look at the plates to identify them. And then we have a very common spring species. This is one of the main actors during spring, and is also one of few, maybe it's just the only one in the Baltic that are making colonies too. This is quite unusual that it's 3 cells in the colonies. Usually they are arranged 4, 8, 16, 32, 64 cells together, and it's quite fun to see them moving around a big colony when all the flagellas are moving. And this is what the single cells can look like. Especially at the end of the bloom, you can see more of the spring bloom. You can see more single cell species. But it's common that they are arranged in colonies, and usually 4 and 4 groups. And this is one of the main actors and important for the biomass during the springtime in the Baltic. Some more dinos. We have this Protoceratium. This is just one species. And we have one called Pyrophacus horologicum. I took this because there are some of them occurring now in the samples. You might see them, and it has special arrangement of the plates here. So this is what's using for identification of the species. But in the monitoring, you usually see them like this. Then it's not that easy to identify it. But there are not that many in that size, and having this row here, then you can identify it by that. But you really would like to see the plates to be able to identify it. Then we have the Haptophytes, or the class Chrysophytes. there. They are small unicellular flagellates, nanoflagellates. But we have this group, Chrysophytes. It's Susanna Haidio has been working a lot with these. She's the one that was working with phytoplankton at our department before, and these were her babies. And they can be toxic. Maybe you've heard about a great bloom in 1988 on the Swedish west coast, that there were a lot of fishes and also seals and other animals had problems, and it seems that it was caused by this Chrysophytes porolefis, one of these groups, and it can be very toxic in when you have high abundances of it. And actually this bloom started the real more heavy monitoring of phytoplankton, and so this helped after this, because they didn't have much information channels from research to the society. So after this bloom, there were 3 institutions started at the county administrative board in Stockholm, on the west coast in Gothenburg, and also in Umeå, to give information to the general public and other things when there are blooms like this or something else going on in the marine environment. So this bloom started that. So it's good to have some information channels, because there were a lot of people phoning different researchers, and they were telling, one said one thing and one said something else, and there was a bit of confusion. So it was very good to have some special places to have that information. And but this has before been called Chrysochromolina, this most common genera, but this class is under revision. There is a lot of different groups going on. So we have, in the monitoring, we have thought about just using either the class Primnesiophyce or in different size classes until it's difficult. Because in monitoring they're small, it's very difficult to distinguish between the different species. So then you have to, and the morphometry between the new genera that they are using, it's not new genera, but they are dividing them into different genera, and it's very difficult to see the morphological differences in between them. So we'll see what happens with this in the future, but there's more molecular studies being done with this. And then we move over to another important group, the Chrysophytes, the heterotrophs, and they are brown, green, yellow-green in color. Never have really green, clear green colors, but it's due to their accessory pigments that they have that give them a bit more of this yellowish-brown color. And in this group, we have what's one of the most important groups in the Baltic, and that's the Diatoms. And also we have some other classes that are belonging to this group. So it's the golden brown algae and this Dictyocophyce. It's just occurring in the southern part of the Baltic. It's mainly a west coast species occurring in Kattegatt and Skagerrak. And we first look at this Chrysophyce, the golden brown algae. They are usually very small cells, 2-3 microns to 10-15 microns in length, and they have 2 different long flagellas. Usually, a lot of them, they're either solitary or like this one, or building large colonies. These can be quite big, and they have this cellulose or chitin lorica that they are sitting in, and they have. It's difficult to take a good picture of them. I've tried a lot, but I had to stop trying because it was difficult. But they have one flagella in this direction and one that are in this direction that are attaching the cells to this lorica. So it's keeping it in the cells. And they have usually are producing special cysts that can be used for identification. And I met a colleague that working at the quaternary, working more in sedimentary ecology and looking at diatoms in the sediments, and he found a lot of small round balls that he wondered what it was, but it seems that it was cysts of these type of the Dinobrium that we will look at. And they are occurring in the Baltic mainly in late spring. And the most important groups here is this Dinobrium and Euglena. And there are two main species too in the Baltic. We have this Dinobrium facilifrum. It's a solitary species. It has this lorica with one cell and a very long spine in one direction. So it's none of the others look like this, so it's quite easy to identify. And then we have this Dinobrium balticum, and it's producing quite large colonies. And for identification, you have to look at the form of these lorica, how pointed this end is, if it's straight here. It's not always easy to identify, but this is the most common species in the Baltic. And then for another Chrysophyte is this Euglena. It's also producing large colonies, also late end of spring, early summer. They can be very abundant. We had a lot of them this year, late spring, early summer. I don't think they are occurring all yet, but we might have them in some of the samples I brought with me to see. And in preserved samples, they are usually breaking up these colonies, so you just see the small flagellas that they have, all the single cells, and then they can be very difficult to identify. So then it's good to have net samples to look at the bigger colonies, and if there might be some left, and instead of just the solitary cells. And another group within the Chrysophytes, is the Pedinales, and we have two genera that are common, Apedinale and Pseudopedinella. This Apedinella has just one species in the Baltic, as we know. Pedinella radians, it was called spinifera before, I think, probably because it has spines like this. And if you see it from above, you can see the chloroplast. It has 6, and this chloroplast is divided like a flower from above. And also this Pseudopedinella, there are a couple of species in the Baltic. The two bigger ones have also 6 chloroplasts, and if you see them from above, you can see the 6 chloroplasts. And it has one flagella in this direction and one in the other one direction. This flagella is usually contracted together and look like more like a lump in this end. And a common species in the Baltic is this small species, Ciliates ficostata. It has just 3 chloroplasts, and if you see it from above, it's difficult to get a good picture, but it has just 3 chloroplasts in it. And one flagella and this contracted flagella in the other end. And they are occurring mostly during summertime, but Ciliates ficostata seems to be occurring all year around, but more common coming in the spring. During summer they are common, and somewhere in autumn they are decreasing again. And then another this class that is more common on the west coast and also found in the southern Baltic, this Dictyoca. And heavy blooms of these can affect fish farms, that because due to these spines they can be attached to the gills of the fishes. So heavy balloons of these can also be a problem. But usually they on the west coast in Denmark they have had some problems with that. And these groups are mainly freshwater species, so they can be occurring sometimes in the Baltic, but they are not common. And then we go to the most important part of the Chrysophyte, and that's the Diatoms. And they are there are many different types of diatoms in the Baltic, and they can be unicellular or colonial. And what's special with these that they have these siliceous cell walls, and they have two frustles, upper and lower part. The centric ones, the one that are circular in form, they look more like a petri dish together with one big, like the lid, a bigger part, and the smaller part, the bottom part of the petri dish. And then we have the pinnate group. They are more like straight filaments. And the diatoms are a very diverse group and important groups. And during spring times we have usually heavy balloons of different types of diatoms. We will be looking at them later on. And they're all heterotrophs, and they have these golden brown chloroplast with these phytoplankton pigments that makes, if you're taking a net sample in spring times, the sea can be totally brownish, and it's used to the color of these diatoms. And somewhat they can regulate their position in the water by vacuoles that they have. Usually some big cells can have really big vacuoles and not that much cell material inside, but the big vacuoles, and they can be using that for regulating. And also, as you will see, they can also have long spines. They can stabilize them in the water column. But since they have these silica frustles, they are usually heavy, and they need the circulation in the water to stay up in the water. So the big cells are usually occurring in spring or autumn time when you have a circulation in the water column, and they are usually when the water is stabilizing during the spring, they are usually settling down to the bottom, and the diatoms are actually the most important nutrient source for the benthic fauna. So what's occurring in the upper parts is important for the benthic. So and as I said, we have the larger groups of the centric groups and the pinnate diatoms, and I will show you some examples of the different groups. The centric species, the cells can be circular, elliptical, or polygonal, and the valve structure is usually radiating from the center. There can be lines of small pores radiating out like this, and they can be unicellular, or they can, like these, or they can form colonies, also in filaments, or together with gelatinous threads together. And they're mainly, the centric species are mainly marine or brackish species. And there are a few of them occurring also during summertime, but that's usually small species. And this is our groups. And we'll look at some of the most common genera. They have this actinocyclus. What's special with this, you usually see like a halo around the cells, especially if you're using phase contrast when you're looking at the same samples. So that can be used as identification of that group. We have this Ateia, for example, Ateia septentrionalis. It's a small cell with long spines, and these can usually be found on other cells that they are attached to. especially Chitosaurus species that I'm going to show you later on. So at late spring they can be very abundant on other diatoms. And they are small, this is just 10 microns, these can be quite big, up to 50, 60 microns. And then we have large genera, these Chitosaurs, that can be a complex group that is not always easy to identify the different genera. I'll show you a couple of them that are more common. These are one of the two larger Chitosaurus species, Chitosaurus impressus and Danicus. This one is usually in longer chains, but they can be single cells too. What's special with this is you can use the name impressus to remember. Usually if you put a thumb on over the middle of the colony, they are going down a bit like that in the middle, so a bit contracted, all the cells are arranged like that. But these are more straight, you cannot see nothing going down towards the special cells for the seta here. So these were abundant last week, so I think you will have them, they might be in the live samples too. So they are common. And also for identification of the Chitosaurus species, you can look at the angle, how the spines are going, how large this area between the cells is, what it looks like, what this window looks like, and the form of the cells, and if there are small spines on the spines and other things. So there are a lot of different species, and I don't know all of them either in the Baltic. But there are some that you can identify to species. This is one of the spring-centric diatoms. It's a very small one, Chitosaurus tronseni. It's just a couple of microns large, and it has just two spines, and they are both on the same side of the cells, so then it's easier. Compared to this Chitosaurus subtilis, it's more a spring species, but it can be occurring in the summertime. And all the spines here are directed backwards, but they have spines on both sides then. So if you just see one cell and you have spines on both sides, then it's this subtilis. But if you just have one side, it's tonsillis then instead. More Chitosaurus species is a common species in the Baltic, especially this one can form heavily blooms if you have very high nutrient areas. We have it in Himmerfjärden here. If they are occurring during spring times, they are having heavy blooms both in spring and also in autumn sometimes, when there are more nutrients coming up from after the mixing of the water. And the special for that is that it has a special form of the seta that they are doing. This bending is special for that, for identification. Some more species. This is usually one cell, single cell, so and has a special form of this, so it can be identified. Species. Then some more centric. These are all centric, so this type of the petri dish type of diatoms. These you can see more easily that it looks like a petri dish. Here you have the upper and lower part, but this one is what's special for that one is that it is not the same size on both the right and left side of it, or whatever it's like, left and right. And also what's special for this species, because you know the Viscus grani, is it has some special area here with larger pores, or of course structures in the middle, and also that you have this structure radiating out. And this species, because you know Viscus grani has been occurring the last years in autumn in quite large abundance, so it can also be important for sediment settling to the bottom fauna in autumn. And it seems to be occurring only after years when we have had cold winters. So at least we haven't had it during the time when we had warmer summers and warmer springs. So there seems to be some connection with it. So they have been occurring the last years and also this year, but we'll see in the future what it is. It's common, and it's occurring now, so you you will probably see it. And then we have a very small species. This Cyclotella, there is a lot of different species in it, but we have one species, Cyclotella schoctavacciana. I hated this to spell it every time when I was going because it's too many A's and it took some time just to make out how to spell it. But I think it's named after some Indian person, or not Indian, Native American name in somewhere in. So it's occurring also on the American continent too. But it's quite nice, and what's special with this one is it's undulated like this, and a special form of it. Quite nice, but it's a small one. And we have some centric species that are building colonies. We have this spring species, Melosia arctica, for example. It's occurring also, it can be blooming under the ice, sitting under the ice, attached to the ice, and also occurring in springs when there have been ice in the area. So this Melosia arctica. And then this Skeletonema, now it's called Skeletonema marinoi, but it's maybe more commonly called Skeletonema costatum, but it has now been known that due to molecular studies that this is this species instead of Skeletonema costatum in the Baltic. But this is a very common species. It seems to be more common in spring when it's more warmer spring times. So, and also it can be occurring during summer in some areas, but mainly spring species. And it can form very long colonies, and sometimes the colonies are curled, but they are usually long, straight colonies, connected by small spines in between the cells here. Then we have another group of centric there. These look like the petri dishes again. We have this Thalassocida baltica. This is also a common spring species in the Baltic. The cells are attached by gelatinous threads, and it has special arrangement of the small spines inside of here for identification. But this is one of the most common spring species. And also this Thalassocida levander is also a common spring species, but it's much, much smaller. It looks like a bit like this, but it has also gelatinous threads in between the cells. But it has also some mucilage around it, which makes that small particles are added or found around the cells. So they usually look dirty due to this mucilage. And they can be abundant in spring. Also to be more abundant after cold winters. And then we have another example of another Thalassocida Höglander. It has the rounded edges here are more rounded here. Here they are cut, all the edges. So that's one way to identify that species. And then we have the pinnate diatoms, the Basiales, and most of them are actually freshwater species. And these cells are more elongated in view, like a rod in shape. And they have a lot of rows of dots, stria, that are important for the identification. And they can be either single cells or cells in colonies, and it's a very diverse group. And there are not that many pelagic species. It's more a benthic group. There are a lot of benthic species. So if you're taking a sample here in the harbor, you will probably hit some of the vegetation here, and you will also get a lot of these benthic species. So if you take a sample here, I will not be able to identify because I don't know all the benthic species, because there are a lot of them too. And to look at them, you usually have to clean the shells and just look at the frustulus to be able to identify them. And you also need to have very high magnification of the microscope. And these are the most common genera in the Baltic of this group. Now we'll go through a couple of them. This is important species in spring, Achnanthes taneata, sometimes also called Pauliella taneata, but I think it's more commonly called Achnanthes taneata. And it forms like ribbon-shaped colonies. It's small cells all together like a ladder or like a ribbon like this. And the cells can be coiled, and when they are coiled like this, and you see them from the side, some can think that they look like nodularia threads. But these are occurring very early in spring, and usually also seem to be more common after cold winters when there have been ice around. And so last year we have a heavily bloom of this Achnanthes, so we'll look at some spring samples from last year, we'll see them. Then we have this Navicula. It's not as common, it's more also occurring especially after icy winters. Navicula van Heffen is a common species. It has also ribbon shaped, but the cells are broader, a bit larger, they're rounded on the edge, and they're quite nice to look at. Here you don't always see the differences between the cells. You can see it, but these are a bit brighter and larger, and you can see the lower chloroplasts and vacuoles inside them. Then a type of this genera Flagellaria. It's like ribbon shaped, and they're attached in the middle here between them. It's more common in freshwater and very fresh coastal areas. Nice. And then we have this Astrionella formosa. It's a lake species, but you can find it in the Baltic sometimes, especially in coastal areas. But it looks like, just to see how different the shapes of the colonies can be, and this looks more like a star in colony form. And then we have this diatoma that they can attach the cells and giving them, they're either two or one cells together, and they can build in very large colonies, but they are attaching on the side, and they are not usually, they can have small stars like that, but usually more irregular colonies. More diatoms. We have this Nitzschia. Nitzschia frigida is also a spring species, important in cold winters, and some of these cold species, they can be absent if you haven't had icy winters, but when you have ice again, they will appear again. So if they are dormant in the sediment or something else, it seems to be something like that. Then there are a lot of Nitzschia species, and for identification you should clean the cell and just look at the frustula and see the stria and how they look like. So it's difficult to identify. But these two are identifiable, especially Nitzschia paleosa. You can see at the end of the Cyanobacteria bloom, they are usually attaching to the nodularia colonies. So if you look at an old nodularia colony, it can look like a lot of needles around it, and then it's usually this Nitzschia paleosa growing around it. And I don't really know why they are attaching there. It seems that they are using the nutrients that are leaking out from the nodularia threads, and using that for their growth. And these are a type of other needle-like diatoms. This Pseudonychia. It's a marine group. We have this Pseudonychia seriata, they're a bit broader, and the small one, Pseudonychia delicatissima. I just wanted to show that because they are important on the west coast, because they can be toxic too. So these are a group that are usually monitored for toxicity on the Swedish west coast. And they can form large colonies. They are attaching to the sides, so they can be, or not colonies, but long threads of cells. And some other forms of diatoms. These are also these pennate diatoms. We have this Cylindrotheca. It can be occurring in if you're high nutrient concentration, this species can be very abundant. And if you compare these, they can look very much the same. This is a more newer genera, but the difference between them is that this one has 2 chloroplasts. You can see the division between them, and this one has just 1. So then that's a way of differentiate between those genera. So the different forms, how the chloroplast is sitting, and the number of the chloroplasts, and so can be important for identification of diatoms. Another group, we have the Euglenoids or Euglenoids, and they are mainly unicellular flagellates with 2 flagella, and one of the flagella can sometimes be so short you don't see it. Here you can, in this species, you can see the 2 different flagellas, and they have a eye spot. Sometimes this is not the eye spot, but carrying these can form also blooms. So sometimes we have had it in the Himmerfjärden area. I know that they have been out and monitoring, and some say, "Oh, the water was totally green sometimes," and then but it disappeared just a few minutes after that, because they have these flagellas, so they can be swimming around like a school of flagellates, like a school of fish, but small things. So they can come up to the surface and then dive down again, and so they can be appearing and disappearing, and so sometimes you can see them. And this Cryptophytes can be quite, if you have live samples, they can be quite greenish, so you can see the water can turn green when they are appearing, and then they disappear again. So they are appearing. And they are especially important in nutrient-rich areas, and we have 2 important genera. It's Cryptophytes and Euglena, but Euglena are mainly freshwater species. You can see it if you go into the Taxa archipelago, you will have getting more and more of these species, because it's getting more and more fresh in that area. And then we have the Chlorophytes, another group where the green algae are a part of that. And we have 3 different classes of this group. We have the Chrysophytes, then we have the Chlorophytes with the green algae, and the Cryptophytes. So, more to show the diversity of the Baltic phytoplankton. Another group, we have the Chlorophytes, and it includes a lot of unicellular and colonial or even filamentous algae. And some of them have a flagella and an eye spot, and they have a more rigid cell wall that makes them that they are not like the Euglenoids or the Cryptophytes I showed you before. They haven't got that rigid cell wall, so they can change their size or the form. But these are more rigid in their form, and they have chlorophyll A and B, but they also some accessory pigments. But many of them turn quite green in the water if you see them. And we have one group that is important during almost the whole year around, and they are quite small. It's this Chrysophytes. The most important genera here is the Pyramimonas. And this group, it has both the cell and the flagellas are covered by small plates. Usually, if you have such small cell, you usually cannot see the flagella always, or a very thin one. But in these groups, they appear much thicker, and it's due to these plates that it has around it. And this Pyramimonas has usually 4 flagellas, and it's more flat in one end and then roundish in the other. And they are very common all the year round, and can be forming a large part of the biomass of the smallest cells too, so they can be important. And then we have, these are not, just to show how different species can look, these are not that abundant, but you can have this Pteropsperma, it's more common on the west coast, and it have two different stages that look, one it's called the Ficoma stage, that has this list on the side of the cells, but it's also a flagellate stage, that one cell with long flagella, so it's difficult to understand that this is the same species, so same group, but different stages of it. So, and then we have very small species, this Pseudoscorpheldia marina, and it was difficult to take a good photo of it because it's quite small, just a couple of microns big, but it has two flagellas, unevenly long, one longer and one smaller one. And this species is mostly occurring in coastal areas and more freshwater, so it's not as important. And then we go over to the green algae, or what I think is the most beautiful part. There are a lot of beautiful species in it. Unfortunately, we don't have that many green algae in the Baltic, but the more freshwater you get, the more of these species. So I will just present a few of them that are more common in the Baltic and that can be occurring. And this class includes a lot of several orders, and the form of them are very, it's a large variety of them. And they can be free living, they can be flagellates, and they are both motile and non-motile forms. So, and they are very similar to higher plants in their cell wall structure, and their reproduction can be both vegetative and sexual, and they are autotrophic, especially in the Baltic, and so I haven't denoted if they are autotrophic or not, because these are autotrophic in the Baltic. And here are some of the most common orders and genera occurring in the Baltic, and there are a lot of more of them occurring in freshwater, but I'm not mentioning those now. But if you're working, for example, in the lagoons of the Coronian lagoons and other things, you will have a lot of the green algae and also a lot of cyanobacteria, but also, so there they have very high diversity of green algae. So we have most common. We have these just to see the difference, because before all these were called Cenodesmus. There are usually 4 cells, it can be more cells than that, 4, 8, 16 together, but more 4 and 4 cells together. But this Cenodesmus has nowadays been divided into 3 larger genera. We have this Acutodesmus that has a bit pointed ends of the cells. The Desmodesmus have spines on the side. And this Cenodesmus, they are more roundish and have no spines on the sides. So this is more what's dividing up the different genera. And these are very common. And so to say, the green algae, it's a lot of thing going on of the taxonomy. And we had a person having lectures about green algae a couple of years ago in this phytoplankton group. And after that, we actually showed, what should we actually call them? You just wanted to see green algae SP, because you couldn't really say to a species because there were so many uncertainties going on. So probably a lot of these names and species will be rewritten afterwards. But it's a lot of things going on in this group according to the taxonomy. But it's mainly more freshwater species, but you sometimes found them in coastal areas. Another species that can be occurring in the Baltic is this group of Monoraphidium, and they can be occurring in the whole Baltic, especially this, for example, Monoraphidium contorto. And these are more common in, yes, Monoraphinium minutum. If it's called, actually I want to call it CF Monoraphinium minutum, because after that lecture we had a couple of years ago, I'm not sure if it's Monoraphinium minutum anymore, but at least it's that genera. And another important genera in the Baltic, it's the Ocystis, and they usually have 4 cells. They can be dividing, and they have some envelope enclosing the cells inside. And sometimes you can have more colonies dividing into this. You can see more than 4 cells in it, or just a half colony too, if they are dividing. And these are occurring during summertime, and they can be very abundant at the same time as you have Cyanobacteria. So I've seen a lot of them, so I wonder if they can easily take up the nutrients, so why they are occurring at the same time. But maybe the Cyanobacteria are leaking something that they can use, or I don't know. But they're occurring during summertime, and also when there are a lot of nutrients, they can be very abundant too. And then we have these are mostly more coastal areas, but just to see the variety of the green algae, we have this pediastrum that you can have. These are called synobium, small cells with different forms that are connected together to form the colony. And they have the outer cells in another shape than the inner cells. And here the lengths of these outer cells and the forms of these colonies are used for identification. But this genera has also been split into other genera now, so I'm not sure if all belong to pediastrum, but in that group at least. And this Spherocystus is a colonial green algae that can be occurring in the Baltic, sometimes in the open Baltic, but more in coastal areas. We had it a lot in the inner part of Himmelfjärden last week, and a few of them were found in the outer part, and maybe at this B1 station. But it can also be occurring, but not in that high numbers. And then we have one green algae that is common in forming filaments. It's like a sheet of cells, and with cells in it. So it's this Planktonema lautemorni, and the size of the cells can be different due to the salinity. They're quite small, and smaller here, 2 microns or something in width here in this area. But if on the west coast, they can be 4 or 5 in width. So they are changing due to the salinity too. And then we have some other, also belonging to the Chlorophytes. It's another class, Cryptophytes, and I just took this just to show that there can be other types of green algae. This Elacatotrix, it's usually benthic species, but sometimes you can find it in a pelagic form. And this Colliella, it can be found in the open Baltic, and especially during summertime. And they look much like this Monoraphidium that I told you before. But in this one, it's the cell division is somewhere here across the cells. But in Monoraphidium, they are dividing like this, 2 cells in that direction. So that's what's different between the different. And then we have looked at all the phytoplankton I wanted to talk to you about, but then I will show you one of the important ciliates that we have that are actually autotrophic, or should we say that it's mixotrophic. It's a bit arbitrary if it's usually, because this Mesodinium rubrum, in some parts it has also been called Myrionecta rubra, but I think that is described afterwards, so this is actually more than the more correct name, I think. But it's important in the Baltic Sea, and it has several chloroplasts that has Cryptophycean origin. And if you're having this species together as cryptophytes in a culture, the cryptophytes are disappearing and they are ending up inside the mesodynium, and they are using the chloroplast from the cryptophytes. They seem to be robbing them of that and using them after that for their photosynthesis, and they can be self-sustaining by that. So it's difficult to know, are they eating them or just robbing them of their chloroplast? So if they are totally autotrophic or mixotrophic, I'm not really sure how to say that. And they are common year-round, but they are mostly common at the end of spring bloom, where there can be a big part of the bloom. And also we have noticed that the size of these can be different in different areas due to the nutrient conditions. Here in Asker they're a bit smaller, and at Landsutflek they're smaller, but if you're going to the Himmelfjord, they're increasing in size. So it seems if they have a higher nutrient concentration, they are increasing in size. And they have a cilia, or these cirri that they can be, I thought they looked like this barbapapa with hula skirt or something. But then when I looked at them in the life sample, I was surprised. I thought they were going to swirling around like this, but they are jumping very quickly. If you want to take a photo of them, you have to be very quickly, because they jump like a flea or something. So if we have it in the life samples, they're fun to look at, and we probably have them because they're occurring all year round. And since they have this cilia, they can migrate, and they are also migrating down to the phytoplankton to get more nutrients, and then they can go up again if they want for the photosynthesis. So. And then we go to the more heterotrophic part that can be occurring together with the phytoplankton. And this is a group with colorless and different types of flagellates. It can be in single-celled or in colonies. And these three groups are the most important for the Baltic Sea, and especially this Ebria. And if we look at Ebria, there is one species in the Baltic. It's mainly Ebria tripartita. It has this skeleton inside, siliceous skeleton that divides the cells into three parts. You can see it here. And then you see the nucleus inside. There's two flagellas to swim with. And this is heterotrophic, and you can see it eating. This spring I saw them eating a lot of skeletonema. So they were sitting on top. I don't know if they were eating it from one side to the other, but because you could see one filament of the skeletonema was just up and stretching out from the cells. So it's quite fun to see. And then you have this Bisoeka, and it's usually attaching to one of these Chitosaurus species or other species. So it has some kind of lorica, and you have the cell and some cilia coming, or cilia radiating out like this. And I saw them in the live sample last week, so probably we can see them maybe too in the sample from this year. It is last place that you wrote something. Then you have the color flagellates, and the Chronoflagellates, and they have it's one cell, and they are encircled by this color that they have, and the shape of this and how these look like is the way you can use for identification of them. And they're heterotrophic, and they can be very numerous after the spring bloom, but also year round. And some of the most common, it's not the best species, but this one has, you see the cell, and then it has the color like this here, radiating out, and this can be sitting in large colonies with small cells, and it's not that easy to see. But these can be, if you're looking very quickly, they can look like this Euglena colonies or something else. So you shouldn't be too quickly when you're looking at the cells. You have to look more carefully what it is. But they are common after late spring. And then we have some groups that are not, we don't really know to what group they are belonging to. They are heterotrophic, and it's a bit unclear. They call it inserted cities. It's uncertain where they are belonging. And they are ingesting bacteria and small algae. And these three groups or genera are commonly occurring in the Baltic, especially during summertime when there are many other small species occurring. So Cryptophytes, Cryptophytes, and Helena. So heterotrophic. And if you're looking in phytoplankton samples, you might end up with other things that are not phytoplankton. Sometimes you can see the cord of the vorticellas, this ciliate, so you just see this part, or you just see the head of the vorticella, and then you should be aware that that's not a dinoflagellate or something else here. And you usually, sometimes you see just the cord of it, what's attaching to something. And you can see these small ciliates, they're floaters, they are walking around on the filaments of nodularia during summer times. On the aggregates, they can breathe very, very abundant. They're quite fun to see walking around. And of course, a lot of different types of pollen grains of different species. It can be from pine, birch, or whatever. And also these other types of ciliates, like this with the lorica, Helicostomella, that is also commonly occurring during summertime. And then I have just some, as I said, I have some information of good. If you're more interested in phytoplankton or want to look at more literature, I've listed some of the more useful identification literature. And also the methods that we are using for the monitoring in the Baltic is this HELCOM guidelines. And also some more literature, you can look more at that later on. And also if you want to look at more pictures of phytoplankton, you can look at this site nordicmicroalgae.org, and there are a lot of images uploaded there. In this HELCOM phytoplankton group, we have talked about having an own site with validated phytoplankton images, but it was easier when they started this. We could add a new gallery to their. So we are uploading our validated images to this. But if you just want to see what we in our group call the different species, what we're using for identification, you can click on the gallery's HELCOM peg, and then you will see what we are using for our identification when we're using for the monitoring for the Baltic. So we haven't gone through all the species yet. This year we were taking in photos of Diatoms, and for the next year we'll have the Dinoflagellates, but then we'll have most groups. Then we have to add some more afterwards with it. And if you want to go more and see more of the latest taxonomy and where it's more valid the world around, for algae you can look at this algae base and also worms, the word register of marine species to look at. And here you can see also old names, the synonyms that have been used. They are also mentioned in this. That's what we are working with. But hopefully I've showed you a bit that phytoplankton can be beautiful, but that they are not just beautiful, they can be very fun too to look at. This little creature looked at me when I was working with my monitoring just before I went for vacation. I said, okay, goodbye, see you after vacation. So it can be fun too, don't remember. And they're beautiful too.